Well, we're just after 5:30, folks, so let's make a start. So good evening, friends, folks and scholars. So my name is Dr. Tim Cheek. I am the Dean of Postgraduate Research in the Faculty of Medical Sciences, and I am absolutely delighted to welcome you all to this latest in our series of Insights Public lectures at Newcastle University. Before we begin, I have a few housekeeping announcements, please. So, please can we ask you to check that your phones are switched off or are on silent. Please do that now. Thank you very much. We are not expecting a fire alarm, so if the alarm does sound, please do follow the instructions of the event stewards. Follow them to the car park outside. If you would like to tweet about tonight's lecture, please do so. And for those of you who know how to do this, please use the hashtag InsightsNCL. So on to this evening's lectures. So tonight we are presenting three tales from the frontier of medical sciences research. And I'm particularly proud because our three speakers are all current PhD students in the Faculty of Medical Sciences. You can see their names behind me. They are all here in the front row. So please will you join me in welcoming James Allison, Doyin, Alao and Laura Booth. So, for anyone who is not aware, a PhD is a doctoral training research degree and is the highest level of academic qualification that you can achieve. So the degree normally takes between 3 or 4 years of full-time research work towards a thesis that then describes an original contribution in the research field. So how did James, Doyin and Laura get here? So in our faculty, we asked our PhD supervisors to nominate their most gifted PhD student speakers, and 6 were selected for further faculty training. So all 6 then presented in front of a panel of judges, including staff from right the way across the whole university, not just the Faculty of Medical Sciences. The judges selected 2 winners, which were James and Laura, and then in a public vote, you folks voted for Doyin. So all 3 speakers tonight are all winners of the Faculty of Medical Sciences Postgraduate Public Speaking Prize, and that is why they are here tonight to describe to you their quests at the cutting edge of science. So before we hear from them, I would like to propose 2 votes of thanks. So firstly, to the training team who have guided and supported all 6 student speakers through their training. So many thanks indeed to Duncan Yellowlees and to Gemma Mitchelson and to Richie Hetherington, who is here in the front row, for their time, energy and effort in helping our students. And secondly, grateful thanks too to our 3 student runners-up in the competition. So we all know, don't we, it's tough to miss out at the semi-final stage. So very well done, and thank you to Jack Clark-Corrigal, Ella Dyer and Huda Gopi. And I think Jack and Huda, are you here tonight? Are you here somewhere? Please stand up, folks. Please stand up. So thank you Jack, thank you Huda. Well done. And before the evening ends, please do come down at the end of the evening for the photographs. Okay, thank you, Jack. So on to the presentations. So to help with the flow, we will hear from our speakers consecutively. They'll talk for about 15 minutes each. Please can we ask you to hold fire with your questions, hold them in your head, and then when the final speaker has finished, we will have a separate question session for all three of our speakers. So our first speaker tonight is James. So please do come up, James. So James is a qualified dentist who teaches our dental students, but this evening he is going to focus on his clinical PhD research in the School of Dental Sciences. So thank you, James. Over to you.
Thank you very much. Okay. So as we just heard, I'm a dentist and a researcher in the School of Dental Sciences here. And I want you now to put your hand up if you've ever been to the dentist. Fantastic. Now keep your hand up if you managed to get to the dentist during the height of the COVID-19 pandemic. Fewer hands there. And this was really hard, wasn't it? Lots of people found it impossible to get an appointment. And it meant that millions of people in this country missed out on vital treatment that they needed. And that undoubtedly worsened their oral health. And the reason for that was aerosols. We can see these aerosols here. And aerosols are just particles, in this case droplets of water, suspended in the air. And we can see an aerosol from a dental drill here. What we can see are the big droplets that are visible, but we can't see the smaller stuff that is too small to see. It's tiny and it will fill up the room if allowed to do so. And this was what caused a problem. We were concerned that these aerosols might take the virus that causes COVID from patients' mouths into the air and pose a risk to staff and to other patients. But you might ask, why were we worried? It's clean water, right? And the water that we use in our drills and instruments is clean, absolutely, yes. But the mouth is not. The mouth is a filthy place. It's full of bugs. Even when we're healthy and we don't have an infection, it's full of bacteria, those bugs that are everywhere, all over the planet. They're on our skin, our guts, our mouths. It's also, even without an infection, full of viruses. And there are probably more viruses than there are bacteria in the mouth. And they don't harm us. They just sit there doing their thing. The mouth's full of fungi too, the stuff that might grow some mold on your food if you've left it out on the bench, or that we use as yeast to make beer and bread. But of course, when we do have an infection, whichever bug, whichever microbe is causing it, multiplies massively. Many, many times more of that bug is in our mouths when we have an infection. So the mouth is full of bugs. And you can see what the problem might be. If we're using a drill or a dental instrument like a scaler in the mouth that we've just seen is full of bugs, well, that might go somewhere. Our aerosols that our instruments produce travel out of the mouth, and there's potential, therefore, to pick up these bugs from the mouth into the air. And this is what kept the doors closed during the pandemic. We didn't know how much of a risk this was, and if it was a risk, what on earth we did to control it and make it safe. So these aerosols that our instruments produce are made up of droplets of lots of different sizes, big ones, little ones, and everything in between. But how big are we talking? Well, if you can imagine a day out at the beach, you picked up a handful of sand, you can imagine how big one grain of sand might be. And a piece of sand might be about 100 micrometers in diameter. So that's 100 thousandths of a millimeter, really small. But some of the smaller stuff that we can see is smaller still. You can imagine dusting a surface, dust floating up. I'm sure this is very clean, obviously, been cleaned perfectly. But if you can imagine dusting a dusty surface, seeing that dust float up into the air, that's probably some of the smallest stuff we can see with the naked eye, and that might be about 10 times smaller than that grain of sand. But the droplets in our aerosols are smaller still, less than 5 micrometers, probably most of them. Still, though, bigger than those bugs we just mentioned, and that means those droplets can carry bugs within them. And here we see the problem. When we produce an aerosol, the really big stuff that we could see in that image before, falls to the floor quite quickly. It doesn't really go that far. The smaller stuff, these big aerosols, not the really tiny aerosols, can go a bit further. They can stay suspended for a bit longer, and we can inhale them. They get stuck in our throat, in our nose, and if they've got bugs in them, they can then instigate an infection. But it's the really small stuff that we worry more about, the tiny particles that are far too small to see, that can get much further down into our lungs and cause a nasty infection. And this stuff is invisible, and it will fill up a room if it's poorly ventilated until fresh air can replenish it. So this is what made these chairs empty and meant the doors were shut. It was the uncertainty over these risks. We didn't know what we needed to do. So what did we do? Well, we did what anyone does when they're trying to solve a problem. We put together a crack team. And this involved dentists like myself, but also basic science researchers as well, from lots of different disciplines to help us answer this question. And initially at Newcastle, at the start of the pandemic, we designed a model to test our procedures and work out where these aerosols might go. And we did that in a mannequin, because by using a mannequin rather than in a patient, we could many, many times repeat a single procedure and be very controlled in what we did and quite reliable. And we designed this apparatus initially in one of our labs, you can see here on the top floor of the dental hospital, which goes out to 8 meters in diameter, so quite a large distance. And we can see half a meter, we've got some platform space where we could take some samples from. And this would let us map out where any of those aerosols go to answer those questions we didn't have answers to. So we did lots of dentistry on poor George here, our mannequin. Can't forget his part in this research, couldn't have done it without him. He's got the best teeth in the northeast. They are plastic though. And we did that in lots of different settings around the hospital. And initially we used a dye to simulate some virus, for example, in saliva. And we could detect this fluorescent dye very sensitively and pick up where any of that aerosol, even at very low levels, might go to. So where did it go? Well, if we look down, bird's eye view, on that picture that you saw in the lab with the big rig set up, we can see what's going on here. Right in the middle is George, our mannequin. There he is. We've got a dentist doing some dentistry just sat to the side. And each of these black dots shows where a sampling location is. And the dark blue areas are where lots of aerosol goes. The green areas where some aerosol goes, and the yellow where not much goes. And what you might be able to see is that most of that aerosol is in that first half a meter or so, around where that procedure is happening. Not a surprise. But still not insignificant amounts, so getting out even to 4 meters away in lots of different directions. So our aerosols go far. How long do they last? Well, we looked at that too. Here we used a very sensitive instrument to measure how much aerosol was in the air at any one time. So you can see on the vertical axis here, more aerosol at the top, less aerosol down the bottom. And we measured that over time during a dental procedure and for some time afterwards. And unsurprisingly, lots of aerosol was produced in that grey box in the middle where we were doing some dentistry, a couple of 100 times increase in the amount of aerosol during that period. But what's less obvious is that even 15 minutes after that procedure, that aerosol doesn't settle back down to the level it was before. So our aerosols go far and they last for a long time. But none of that really matters if it doesn't have bugs within it. So we looked at this too. Now, we asked our hospital if we could spray the COVID virus around our clinics, and they said no. Wonder why. But viruses work by attaching to our cells, and this might be the cells that make up the lining of your nose. They come along, they stick on, and they cause an infection. Luckily for us, bacteria get viruses too, just the same, okay? And these viruses are called bacteriophages, which literally means bacteria eaters. They kill bacteria. And we might think that's a good thing, and for us it was, because it meant we could use them without any risk to human health. We could spray them around our clinics with gay abandon. So these bacteriophages, phages to their friends, were used in our model that you saw before instead of that fluorescent dye. So we had these viruses in the mouth of the mannequin simulating some virus we're interested in, be it flu or COVID or whatever else. And then we looked at where that went and where we could detect it. So looking down on that area again, as we did before, where did the virus go? That looks very similar. It's mostly these orange and yellow areas within that first half metre around the procedure. So this colour here corresponds to about 10,000 viruses per square centimetre. Really small area, lots of virus. But beyond that, not much. A couple of virus copies, perhaps. So our aerosols go far, they last long, and they've got some bugs within them. So it's worth saying that this was an absolute worst case scenario. We really wanted to understand, at worst, what the problem might be. So this is someone with a raging infection we're simulating here, lots of virus in their saliva. We're spraying this drill around. We're not using any of the protection that we would use when you went to the dentist during that period. But how do we make it safe? How do we open those doors back up and get people back to having some dentistry that they really, really needed? Well, we looked at lots of things, and one were the drills that we used. So the drills the dentists mainly use are like little jet engines. They use compressed air to drive a tiny little turbine in the head of this drill, and that air has to go somewhere, and it's blasted out the front of the drill with water, producing this really fine mist that we saw before to cool that burr down, and it goes really far, as we've seen. But what if we used an electric drill instead that doesn't use air, that can just squirt water out of the front? You see those droplets are much bigger, so as we saw before, they won't go as far, and that's exactly what we found. Much less aerosol produced, very little at all, in fact. So our drills work. But of course, when you go to the dentist, we use the suction, we use the hoover, don't we, to get the water from the back of the mouth if you're having a filling. But of course, this is catching aerosol too. It's our thought, certainly. And we tested this and found that actually this was reducing the amount of aerosol that escaped the mouth by about 80% anyway. But some aerosol will, of course, get out of the mouth, so what do we do about that? Well, we tested these two. These are big hoovers, basically, or local exhaust ventilation to give them their full name. And you can see what they do. They capture any of that aerosol that escapes the mouth. And we found this reduces it by about 90%, so really substantial reductions in how much aerosol gets out of the mouth. Can we reduce the amount of virus in those aerosols, though? As dentists, for many years, we've used disinfectants in the water lines, in the pipes that supply our drills to stop bugs growing there and stop the water coming out being horrible. And we thought if it's killing bugs in those pipes, in the water lines, well, the aerosol's made up from that water, so surely it's killing the bugs in the aerosol too. And we tested that, and the answer is it is. And we're using this in the clinic anywhere, all over the country. And this reduced the amount of virus that was escaping the mouth in those aerosols by about 96%, massive reductions in the amount of virus in there. And finally, can we separate the bugs from the drill altogether? This is a rubber dam. We've used it in dentistry for well over 100 years. It's nothing special. If you've ever had a root canal treatment before, you might have had it used. All it does is isolate the tooth that we're working on from the rest of the mouth, so that if we're cleaning out the inside of this tooth, bugs can't get back inside it and cause an infection. But of course, that separates the saliva in the mouth from the aerosol in the drill. And that's what we found. When we use a rubber dam, much, much less virus gets out of the mouth, about an 86% reduction. Now, of course, none of these measures are perfect. They all have holes. But by layering them up together, we reduce that risk down to an acceptable level. And I said before that our model was a worst-case scenario. Many of these things dentists were already doing, so that risk is likely to be very, very low as standard. So during the pandemic, this was really helpful for us in Newcastle, but more widely, and the work was cited or referenced in lots of different guidance documents in the UK, and helped open the doors back up to dentistry and also teaching our students on the clinics as well in our dental school and lots of others around the world. But what we really found, as I said before, is the problem was the uncertainty. The problem was we didn't know how much of a risk this was. So if we were to see the same thing again in another pandemic, hopefully now we have a much better understanding of how much of a risk this is or isn't and what we do to control it, to mean we don't have to shut the doors again, we don't have to have people miss out on the treatment they need, which worsens their oral health. But of course, as dentists, we will see patients with infection, somebody coming with a cold or with the flu, and most of the time we'll get them to reschedule that appointment. But sometimes you can't. Sometimes it might be an emergency treatment. And hopefully now we know much more about what things are needed to make that treatment safe in a patient with an infection. So things really did feel like they were up in the air at the time. Now that the dust has settled, though, we know much more about these aerosols that we create as dentists every day, how much risk they do or don't pose, and what we can do to control them. So thanks very much for your attention, everyone. Thanks to everybody who worked on the research I've just shared with you and the funders who made this work possible.
Thank you very much indeed, James. Please hold your questions, folks. And I will introduce our second speaker, who is Doyin Alao, as you can see. So Doyin is a qualified GP who's interested in improving education and training for all our doctors. And she's going to focus on her PhD research in the School of Medicine. Over to you, Doyin. Thank you.
A patient once said to me, okay, I'll try it again. Hello? Is it working? Yeah, that's good. A patient once said to me, we'll call him Bill for tonight. He said, I was born in the days where you do what the doctor says, no matter what. He was describing an experience to me whereby he was having a procedure done and it was really hurting him, but he sat still and said nothing because he felt he shouldn't. This signifies the type of relationship doctors had with their patients over 80 years ago when he was born. How many of you have consulted with your doctors before the year 2000? Many hands. How about before the year 1980? Fewer hands, thank you. I would like to take you down memory lane. I want you to remember how the consultations used to go many years ago. How much did you get to say when the doctor was discussing what treatment options you could have? And how many options did the doctor give you? I bet it's fewer or no option at all. It was just, this is what you have to do, and then you go and get the medicines and use it. However, the relationship between doctors and patients have evolved massively over the years. And patients come to the doctors having an idea of what might be wrong with them, because they might have spoken with family, they might have spoken with friends, or even Googled it. So nowadays, doctors work hand in hand with patients to come and develop a plan that is suitable and acceptable to the patient. And it means patients are happier and they're more likely to follow through with their plans. Now, if we want our future doctors to be better equipped to work hand in hand with patients in this way, we need to ensure that they have good quality interactions with patients during their training. And this is what my project was about. Hello, everybody. I'm Doyin Alao. I'm a GP and a lecturer at Newcastle University, and I'm super pleased to share the results of my research with you today. Every year in the UK, we have over 8,000 new doctors that graduate, and these doctors will spend many years of their lives treating millions of patients, and that means they're keeping us healthier and living longer and living happier lives. Now, in order to become a medical doctor, students have to train in university for usually 5 years, during which time we call them medical students. These medical students interact with patients during their training because they learn about diseases, they learn how to relate with patients, and some of those interactions take place in general practice surgeries, where students may sit down in consultations, observing the consultations with the GP, or sometimes students may consult with the patients before the GP assesses. And those were the patients my research was focused on. My research was focused on how we can enhance the active involvement of those patients that attend general practice surgeries to make sure that they're not there as objects through which the students learn. They have a wider role and more actively involved in the students' training. So what I did, I conducted questionnaire surveys with patients and spoke with patients as well about how they feel about participating in students' training. And then I spoke with students about what they learned from that patient-student interactions and how to enhance their learnings from there. And then I conducted a workshop with patients, students, and doctors to find out how we could address issues that were identified from the research and enhance patients' active involvement. I found out, reassuringly, that patients are generally happy to take part in medical students' training. 4 in 5 patients would agree to participate. And many times they would agree because they want to help to develop the future doctors. They want to give something back to the NHS, and they also want to help the future patients as well. They also identified some benefits to themselves. They felt that they could get longer appointments by seeing the students. They could get more thorough appointments because the students have more time, and they are more thorough because they're just learning and they want to do everything properly. Some patients also said they enjoy hearing GPs teach, and by doing so, they learn more about their illnesses as well. However, some patients were worried about privacy. Some were also concerned about student skills, that because students are still learning, they might make mistakes, miss important diagnoses, or even sometimes they might have to get procedures repeated, which could be uncomfortable for patients. Regarding their active roles, patients demonstrated that they could perform wider roles than being an object. And a few of them actually performed some other roles. Examples were, some patients explained their medical conditions to students, numbers and how they manage their conditions. Some of them told their stories as well, what it feels like to live with those conditions. And some patients even reassured the students when they appeared nervous or anxious, and that boosted the students' confidence. And some patients provided feedback to students about how they're interacting with the patient and how to improve that. The interesting thing is that students found it very valuable when patients performed these roles. They loved hearing about patient stories. Everybody likes stories. So it helped the students to remember things that they've learned, but it also helped them to appreciate how it feels like to live with those illnesses, which means that it could help to develop their empathy. Some students found it nerve-wracking to do things for the first time, interacting with real patients in the clinical settings, and having the patients reassure them was useful for them. It encouraged them to perform those roles and also want to do more. And very importantly, students really valued feedback from patients. They wanted to know how patients feel about their interactions with them and how they can improve their approach to managing patients and speaking with patients so that it meets the patient's expectations. However, many patients did not have the opportunity to perform these roles. It's only a few of them that perform these roles. And the patients in my focus group said they really wish they can do more than just being there. They want to perform many of these roles that they've mentioned. Regarding feedback, one of the patients in the focus group, I'll call her Sue tonight, said, I don't think I would even think of doing that in the doctor's surgery if there was a student there, unless we'd been asked to. So basically, she's saying she can give feedback normally, but if she's in the doctor's surgery, she wouldn't give feedback to students unless she's invited. And what that meant for the students is that it means a huge learning, they've missed a potentially huge learning opportunity to learn more from the students. So the question is, how can we enable patients to perform these roles? So first, 3 things are important here, and the first one is raising awareness about medical training. Interestingly, only about half of the patients in the survey knew that medical students are training to become doctors. Also, only about half knew that medical training takes about 5 years in the university. And what that means for interacting with patients is that, interacting with students, is that if you don't know who is actually in your consultation, it's going to be hard to interact with them to have a rapport with them. And it's also going to be hard to know, what they're learning, how they're learning, and how to contribute to that. So here I am today, and that's why I'm happy today to be telling you about medical training. If you want to know more, I have my email at the end of this session. You can send me an email. I might organize other events where you can interact with students. The second thing is managing consultations. Remember the first quote I gave about Bill at the beginning that shows the traditional doctor-patient relationship. That relationship, whereby doctors are at the top of power hierarchies, and especially where that power hierarchy is steep, makes patients feel uncomfortable to participate in training because they don't know what they are allowed to do, and they don't want to step out of their boundaries or what they feel their boundaries. So if we want to encourage patients to actively participate in training, it's then important to manage that consultation in a way that makes patients feel comfortable and makes them feel welcome to participate in that dialogue that takes place between the doctor and the students. The third thing, which I think is very important here, is enhancing or improving the processes that patients experience when they're involved in training. Now, these processes can include how we invite them to participate in training, how we seek their consent, how we even say thank you to them when they finish interacting with the students, and how we invite them to give feedback to the students. I would spend a little bit more time on consent, on seeking consent. Now, some people might be thinking that it doesn't matter. What does it matter if we don't seek consent properly? I think it matters because imagine this is a good weather now. We'll start having barbecue parties. Imagine organizing a barbecue party and then somebody comes to your party without being invited and you don't know that person. That can make you feel awkward, and it can even make that person feel awkward as well. Now, that is similar to medical training. If patients have come for their consultations and they expect to have a consultation with their doctor alone, if they have a student in there that they didn't know was there and they're not happy to have them there, it can make both patients and students feel awkward during that interaction, and then unlikely to interact or relate with each other, and also make negative experiences for them. A student said, I'll call her Sharon tonight. She said, I've felt personally as a medical student, sometimes I'm uncomfortable if I don't feel there's been proper consent. If I don't feel a patient has the option, it doesn't feel as interactive. So what that means then is that it's unlikely if the student doesn't feel, feels awkward, or the patient feels awkward, they're unlikely to interact, and they're unlikely to learn all that. The students are unlikely to learn all those huge things that we talked about before. So the question is, how can we enhance patient autonomy? Again, 3 things. The first is giving them some time to think. Sometimes, if we ask patients to take part in students' training and want them to give an answer on the spot, it can make them feel under pressure to accept. So sometimes they need some time to actually weigh their options and consider whether they really will want to participate. And that's why participants have said it's important to ask them early, maybe when they're booking their appointment. The second thing is giving them multiple opportunities to decline. I just think about it like this. If we want to spend huge amounts of our money on something, like taking out an insurance, even if we've said yes when the nice salesperson has pitched to us, when we go back home, we start to think about it, do I really need it? It's then important to then give them a cooling off period so that they can say, actually, I don't want to do this again. Again, that's related to medical training. Sometimes patients might have said yes when they have booked the appointment or when they've been invited. but when they think about it, they really don't want to do it, or their situation, their illness might have changed. It might have become more serious, or they might be worried about it, and they want to opt out. So it's then important to give them some more opportunities to opt out. So we could ask them again when they come for the appointment, or when we're walking them into the consulting room, or even check again when they're inside the consulting room with the students. Now, the final thing is recording patients' preferences in the case note. Now, usually we ask patients every time they come, every time we want them to take part in training, whether they want to participate. Now, sometimes if we can think about a system whereby we ask all the patients in the surgery about their preferences to take part in students' training, and whether they would like to participate, whether it's contingent upon their illness or what the grade of the year of the student, it might then mean that we don't have to be asking the same questions over and over again. Because that means that sometimes if we ask the questions over and over again, it can be annoying to patients, it can put them under pressure to say yes. But on the flip side, if we ask everybody in the surgery, it then sensitizes them to the fact that teaching takes place in that surgery, and those that have not even thought about it before can actually volunteer to take part in training. And that means that we have more patients involved, they're happy to take part and they're ready to take part. It means it's better experiences or more positive experiences for them. And that's exactly what we want. My take-home messages are that patients can provide valuable learning opportunities to medical students by performing active roles, like explaining, reassuring students, and giving feedback. We can support them to do this by enhancing their awareness about medical training, improving the processes of involving patients, and managing the consultations effectively. Very importantly, inadequate consent can lead to negative experiences for patients and students. So if you are invited to take part in medical students training and you don't feel up to it that day, it's okay to say no. And it's also okay to share your concerns or what your preferences are, because we want to make it a positive experience for you. Many thanks to my supervisors, Jill, Brand, Susan, and Hugh, and Roger, as well as the patient advisors, Steve, Caroline, and Terry, for all their support with this project. I have shared my ideas with you. I'd like to hear your ideas about the priorities, what are most important in improving patient-student interactions. So I'll invite you to fill a survey. It just takes about 3 minutes. I've left the form at the reception as well, so you can scan the QR code. Thank you.
Thank you very much indeed, Doyin. So our third and final speaker is Laura Booth. So Laura is training to be a toxicologist, and she is in the final year of her PhD studies. So Laura won the university's 3-minute thesis competition in 2022, where she won both the Judge's Choice vote and also the People's Choice vote. And Laura was one of 67 institutional winners across the whole country who were whittled down to 12 semi-finalists and then eventually to the final 6 in the national final of that year. So, over to you, Laura.
Everybody hear me okay, just to check? Fab. Hello everybody, I'm Laura. I'm going to be speaking to you today about my PhD research on cancer chemotherapies and their side effects that they can have on patients' hearts later in life, and whether these chemotherapies are really the hero or the villains of these stories. So, I'm going to start with a slightly depressing statistic, but I promise it will get cheerier from then. Cancer Research UK is now telling us that the lifetime risk of cancer for British people is 1 in 2. But on a much happier note, 1 in 2 of those who are diagnosed will survive that diagnosis for 10 years or more, which is fantastic. So we know that we have a growing cancer survivor population on our hands. Now, this growing cancer survival rate is due to lots of different things, like better awareness of how to spot the signs of cancer earlier in the public, better screening for cancer, and also a plethora of really effective treatments that are available to our doctors. One example of these is anthracyclines. So what are anthracyclines? Anthracyclines are a family of life-saving chemotherapies used in the clinic. They've saved millions of lives, in the 50 or so years that they've been around. There are 4 main drugs used in the clinic. The one that I'll be speaking a lot about today is doxorubicin. So doxorubicin is used to treat loads of different types of cancers, notably leukemia, lymphoma, sarcoma, and breast cancer are some common ones. It's a really, really fantastic, amazing chemotherapy drug. But a lot of us in the room will know somebody, perhaps a loved one who's received chemotherapy, maybe received chemotherapy ourselves. The words fantastic, amazing, not typically associated with a chemotherapy journey. You might remember how exhausted or in pain a loved one was on chemotherapy. I remember how my mum's hair fell out and she was devastated. She was such a glamorous woman. It was a really hard journey for her. And the sad fact is that although these chemotherapies are really potent drugs and they are cancer-beating, life-saving, they unfortunately do have side effects. It's a little bit like when you go to the cinema and you go and see the latest superhero film and you go and see Superman fending off the latest, greatest baddie. And the backdrop is Metropolis or New York City. And when the battle is done and the goodies have won and the baddies have lost, the camera pans. And as the dust is settling, you see that skyscrapers have fallen, cars are on fire, the city is in ruins. And this is the price to pay, apparently, for saving the world. This is a little bit like what your body goes through during chemotherapy. We experience collateral damage in the fight against cancer. So doxorubicin is often known as the red devil chemotherapy due to its bright red colour, but also its notorious side effects. And one of the nastiest of these is that this is linked with long-term heart problems for cancer survivors later on in life. So the timeline might go something like this. A patient will receive doxorubicin chemotherapy. Their heart experiences an injury in the short term. And over time, that patient's heart becomes gradually worse, but the patient won't experience any symptoms straight away. Over time, things get worse, and eventually the patient will experience heart failure symptoms. So in a sense, these patients are really ticking time bombs for heart failure later in life. But it's important to note that not all patients who receive this chemotherapy will develop this so-called cardiotoxicity. Estimates vary, but it's thought that anything from 5 to 37% of patients will develop these heart problems after chemotherapy, and that estimate depends on lots of different factors. So it's important, and we can see that the doctors who are treating these patients have a really difficult balance to strike here. We have the benefit where we need to give our patients enough chemotherapy to treat the cancer and stop it from coming back, but also balance that risk that more chemotherapy might be associated with the risk of heart disease later in life. So really tricky clinical decisions to navigate. So how does doxorubicin, this chemotherapy, how does this work on a molecular microscopic level? Well, essentially, doxorubicin damages the DNA of our cells, the building blocks of our body, and that causes breaks in our DNA of our cells. Importantly here, doxorubicin is stupid. It's a molecule. It's not smart. It can't discriminate between healthy cells and cancer cells, so our healthy cells often take a hit at this point as well. Now, for a cell, the DNA is kind of like an instruction manual. DNA tells a cell what to do, where to go, even who to be. And when there are breaks in the DNA, it's kind of like half the words out of that instruction manual go missing. All of a sudden, the cell doesn't have rules that it can follow, and it might make mistakes, which leads to disease. So this sets the internal alarms going in a cell. At this point, the cell will have a decision to make, and it reaches a crossroads. And there's a few scenarios here. The first is that the cell might die. It might realize that it can't repair this DNA damage, and it's actually better off than rather than risk it, try and follow these rules that it can't understand and might cause further disease. The cell actually enters voluntary cell death, and this is kind of what we want to happen to our cancer cells. In another scenario, the cell might use sophisticated machinery to repair the DNA damage. The cell will survive, and the alarms are turned off. But in a third scenario, and kind of the worst of both worlds, the cell won't be able to repair its DNA damage. The cell survives, and the internal alarms are still sounding full blast. And this causes the cell to enter a zombie-like state called senescence. So what is senescence? Well, senescent zombie cells, sometimes called, these cells stop dividing forever, so they can't make more of themselves, for instance, to heal a wound. They also have a very dysregulated, strange metabolism, and they have a swollen nucleus, which is kind of like the brain of a cell, which I'll come on to later. They belch out harmful, horrible substances into their surroundings, which promotes inflammation and disease for the environment. This can also influence cells around them. Importantly, they resist dying. They don't do the decent thing and go through this voluntary cell death process. Instead, they stick around and they cause trouble for the healthy cells around them, sometimes causing them to become senescent as well. So these senescent cells, they're a real pain in the neck, and they've been linked with lots of different types of human disease recently. So what's the big idea? Bringing all of this together, our running hypothesis was that doxorubicin chemotherapy causes heart failure because it makes heart muscle cells senescent via DNA damage in the first instance. These senescent heart muscle cells then go on to cause problems for their environment, and this encourages heart disease over time. Now, I'm not allowed to do experiments to address this question on real people, rightly so. My day-to-day looks a little bit more like this. So I work in a lab and I grow cells in plastic dishes and do experiments on those. So when we were designing our experiments, we wanted to be mindful of the way this plays out in the clinic. So as a reminder, a patient will receive their doxorubicin chemotherapy. Months or years will pass and the patient won't experience any symptoms until much later on. The patient will then develop heart failure, and we're really interested in what happens here, down this end. What's changed? So we used this to inform our experimental design. So I got some human heart muscle cells in a dish, and I dosed them with doxorubicin chemotherapy for just 3 hours, so not very long. I then took all of the drug away and let the cells just recover for 10 days. And at the end of that 10-day period, then I checked to see what had changed, what's different. And I took pictures of the cells under the microscope, and I also had a look at their internal messaging system to see what's changed. So the first question on our minds was, can this chemotherapy actually make our heart muscle cells senescent into zombie cells? And we have a small checklist that we can look for to try and answer this question. We looked for these two main proteins called P16 and P21. Essentially, these proteins tell a cell to stop dividing, which is a really important characteristic of zombie cells. So as I say, we took these cells, we looked at them under the microscope, and we're able to paint the different parts of a cell that we're interested in by different colours under the microscope. So for instance here, on the left-hand side, let's take this image first, we can see lots of blue circles. These are the cells laid flat on a surface, and each blue circle is a cell nucleus. In green, we have this protein that we're particularly interested in called P16, which is produced inside the nucleus. Now on the left-hand side, these are cells which haven't received chemotherapy. And you can see a little bit of green staining, perhaps a little bit around here and up here. But importantly, not a whole lot laid on top of the blue circles here. However, when we look at cells which have been treated with chemotherapy, which are on the right here, we can see those cell nucleuses, the blue circles, they light up like a Christmas tree, right? We see this really interesting green spotty stain, and this is that P16 protein, which we do see in cells treated with doxorubicin. Our other marker that we're interested in is P21, so different marker, different colour this time. And we can see again, on the left-hand side here, there is a little bit of red staining here, particularly around here, inside those cell nucleuses in blue. But on the right-hand side, we do see a good deal more, we think, in the cells treated with doxorubicin. But because we're scientists, we love to put a number on these things. So we quantified these images, and the numbers did hold up. We saw more of this senescence checklist of P16 and P21 in cells treated with chemotherapy, compared to those treated with no chemotherapy. So that was all well and good, thinking about this green marker, this red marker, P16, P21. Let's just strip it back to those blue circles, those nuclei that I mentioned, which are like the brain of a cell. We also noticed that at a very basic level, the cells that were treated with doxorubicin had larger nuclei, swollen nucleuses. And this was really quite notable, and this is another hint to us that these cells have become zombie-like or senescent. So, we used our small checklist to see whether these cells have become senescent. The next bit of data I'm going to show you is a little bit different. We hopefully have answered our question using our small checklist that yes, these cells have become senescent after chemotherapy. But we wanted to open up our approach a bit, cast the net wider and think bigger. So we decided to examine the internal messaging system of these cells. Now, a cell's DNA lives in its nucleus, but that's used to make instructions which are made out of RNA. And this is essentially like a cell's internal messaging system, which is sent to other areas of the cell and further beyond to respond to changes in their environment or stress, for example. So it can be really useful and helpful to look at changes in the RNA when we're trying to answer some of these questions. Now, this was a lot of data that came back to us, and it came back to us in this horrible graph here on the right. But essentially, what we saw is that about 500 different genes were different after chemotherapy treatment. Every dot on this plot is a specific gene. If the dot is in blue, that means this gene went down after chemotherapy. And if the dot is in red, this is a gene which went up after chemotherapy. So when we looked at this data with the knowledge that we already had, we saw some familiar genes there. We thought, we know a bit about this, we've seen this one before. But because there was so much data here, we also saw some novel, brand new genes that we'd never actually seen before, which was really exciting for us. So by casting the net wider, we were able to see that lots of genes are different in human heart muscle cells 10 days after they've been treated with chemotherapy. So let's recap, and what do we do with this information? So as a recap, some chemotherapies, like anthracyclines, cause heart disease later in life. We think that a process called senescence is partly to blame, where heart muscle cells become zombie-like because of DNA damage in the first instance, and that encourages heart disease over time. We're starting now to paint a picture of what these zombie heart cells are like. So we've got all of these lovely pictures, and the green marker and the red marker and the blue nuclei, and then that dot plot that I showed you at the end. But what does it really all lead up to? The key here is that we're trying to know thine enemy, right? The more we know about these senescent heart muscle cells, the more we might be able to figure out how to target them and what makes them tick. And ideally, how we can fight them, like Wonder Woman here. So in the near future, we're hoping to explore a few different things about how we can tackle this. The first is that, can we kill the zombies? i.e., can we develop medicines to specifically kill off these senescent cells? And there are a class of drugs that are coming up through research at the moment called senolytics, which do just that. The second thing is a little bit more nuanced. So your human heart muscle cells, they're pretty special and they're quite precious. Our bodies don't make very many of them over the course of our lifetime, and they don't get replaced very often either. They're very precious. Not so much like our skin cells, which are manufactured and replaced fairly often. So maybe it's more important to hang on to those cells. And can we think about curing the zombies? Can we develop medicines to change or reverse the bad aspects of these senescent cells? And there are some interesting studies coming through the labs now looking at this class of drug called xenomorphics, which do that. Thirdly, doctors still find it quite hard to predict which patients are going to develop so-called cardiotoxicity after chemotherapy and which ones don't. We're hoping that we could use some of the ideas that we've explored here to possibly inform on that and keep a closer eye, for instance, on patients who develop more zombie cells in their heart after chemotherapy, so we can follow up those patients' heart health more closely. So, to sum up, the big question, chemotherapies like doxorubicin, hero or villain in the end? Well, I would say hero. These chemotherapies have saved millions of lives over the past half a century, but nobody's perfect, not even Superman. So by researching these flaws in these medicines, like how these chemotherapies can cause senescence in some cells, we can hopefully prevent today's cancer survivors, of which there are increasingly many, from becoming tomorrow's heart failure patients. So I'd just like to thank my supervisors, Jason and Gavin in particular here, my colleagues here at Newcastle, my funders, and of course my friends and family for supporting me the past 4 years. Thank you very much.